Down at the Scripps Research Institute in La Jolla, California, researchers did a unique study to learn about how binge drinking affects an adolescent brain. Traditionally, such experiments are done on rodents instead of humans due to legal restrictions. However, this particular study used non-human primate models like baboons and different types of monkeys. These primates are genetically very similar to humans, show many cognitive abilities, and readily consume alcohol to the point of intoxication. In addition, the subjects of this experiment and humans share a similar length of adolescence. As far as we are concerned, these primates are as close to teenage humans as possible.
There were two phases of the experiment, an induction phase and a maintenance phase. All animals went through the induction phase, which consisted of 40 drinking sessions. The animals were then broken up into control and variable groups. The maintenance phase consisted of 200 sessions over 11 months. During this phase, the alcohol group consumed alcohol and the control group consumed vehicle solutions of equivalent volume. Blood samples were drawn on 2 occasions to determine blood alcohol levels. These levels increased as the function of alcohol consumed.
Over 2 months after the last session of the maintenance phase, they removed the animals' brains, froze them at extremely low temperatures, and examined the hippocampal sections from the control and alcohol groups. They were specifically looking for changes in cell proliferation and immature neurons. This is what they found.
All findings add up to a simple statement. Exposure to alcohol increases neural degeneration and decreases hippocampal neurogenesis in the adolescent brain.
So, consumption of alcohol not only deteriorates neurons, but also prevents them from being made. But it's hard to realize what this means in our lives. So we consulted a neurologist who voiced her experience with patients who have abused alcohol.
So the doctor we talked to began by validating the findings of the previous experiment. She said, "There's very clear pathological evidence of damage, including microhemorrhages and atrophy, which is shrinkage in the brain from chronic alcoholism." She then talked about 2 related diseases, Wernicke's encephalopathy and Korsakoff's syndrome. Both stem from vitamin B1 deficiency.
Alcohol provides a tremendously large amount of calories. Most alcoholic drinks are made by fermenting and distilling natural starches and sugar, so it's easy to see why 1 glass of wine is said to have as many calories as 4 cookies. Moreover, the calories in alcohol are relatively useless and devoid of nutritional value. A problem arises when one gets a large portion of their calories from alcohol and not other sources. This causes a vitamin B1 and thiamine deficiency and therefore lesions in the central nervous system.
That's what Wernicke's encephalopathy and Korsakoff's syndrome are. Lesions in the central nervous system caused by vitamin B1 and thiamine deficiency. These diseases are common in developing countries where people physically cannot get nutrients. When people drink a lot of alcohol, they are depriving themselves of nutrients and causing their central nervous system, among other body systems, to suffer.
The neurologist we interviewed testified to seeing people who have vision problems,
Difficulty walking,
numbness,
pain in feet,
loss of joint position sense,
loss of vibration sense,
severe discoordination,
and amnesia. All of these are results of drinking too much alcohol.
On a few occasions, she says people have confabulation, a condition in which they make up stories they think are real.
When you mess around with your central nervous system, nothing good happens.
The chemical structure of alcohol has various ways of making the adolescent brain more tolerant of and dependent on it.
The neurotransmitter GABA illustrates this fact. GABA is a primary inhibitory neurotransmitter in the brain, which means it stops mental processes. When you drink, GABA mediates some of alcohol's sedative effects, but alcohol enhances the effect of GABA on its receptors. So as you drink more, you become more tolerant and more dependent on alcohol.
The GABA system, as with the glutamate and dopamine systems, goes through a rewiring during adolescence. Studies on rats show that the number of GABA receptors in the brain increase dramatically during teenage years.
But The GABA system is only one of the many things that alcohol changes in the brain during adolescence. Alcohol affects many other parts of the brain in the same fashion.
Neuroplasticity is a general term describing the brain's ability to make new connections throughout life. Suppose your dad is a very successful guy, and for most of your childhood, you hear him say that success takes hard work. In your brain, a physical neuron connection forms, making a bond between the idea of success and hard work. Neurons that fire together, wire together.
But then you go to college and hear stories about success being easy. With each success story, the bond between success and difficulty weakens, and the one between success and laziness strengthens. This ability to weaken certain connections and strengthen others is neuroplasticity.
As an adolescent, you make and break millions of neuron connections as your view of the world forms. The strength of a connection between two neurons is based on the speed of neural transmission through the axon and efficiency of the neurotransmitters at the synaptic terminals. Better neurons are insulated with more myelin, which is a special insulation that makes neurons quick and efficient. An unmyelinated neuron is 100 times slower than a myelinated one.
When you hear people say white matter, they generally refer to the myelinated part of neurons. This part is fast like high-speed internet. On the other hand, gray matter refers to unmyelinated synapses and cell bodies. Information doesn't flow as fast through gray matter. It's like dial-up. Unmyelinated brain cells cannot do things that are as complicated as myelinated ones can.
Throw alcohol into the equation and you have a problem. Alcohol destroys and prevents the formation of white matter, which decreases the power of the brain. But it goes further. Neuroplasticity enables new connections to be formed rather easily during adolescence. Under the influence of alcohol, you make the wrong connections.
The bottom line is that until your mid-20s, your brain is remodeling, strengthening, organizing, and growing. Alcohol comes in and disrupts these processes. As a teenager, the world is your classroom. You learn new things all the time. Alcohol is like a distracting handicap that disrupts your learning.
So we know that alcohol increases neural degeneration and decreases neurogenesis. We heard about some real-life experiences a local neurologist had with chronic drinkers. We know alcohol negatively alters the brain at a crucial point in its development. But we still don't know the exact parts of the brain that are affected.
The brain develops in a back-to-front pattern. This makes sense because the rear parts of the brain control basic behaviors that are essential for survival. The brainstem, for example, controls breathing and heartbeat. This part of the brain naturally develops first. The brain finishes developing at around 25 years of age. If you think about the back-to-front pattern, it makes sense that the parts of the brain that are developing in adolescence are the prefrontal cortex, the frontal lobe, and the hippocampus.
The prefrontal cortex and the frontal lobe are thought to be involved in working memory, voluntary motor behavior, impulse control, rule learning, spatial learning, planning, and decision making. Teenagers do stupid things because their prefrontal cortex and frontal lobes are still developing. When you add alcohol to the teenage brain, they have more trouble making decisions and controlling impulses.
The functions of the hippocampus include consolidation of new memories, emotional responses, navigation, and spatial orientation. It all lines up. Teenagers have varying emotional responses because their hippocampus is still developing. Alcohol has been shown to impair the formation of new memories, but it doesn't affect all types of memories the same way. In one study, intoxicated young adults could recall items on word lists immediately after the lists were presented, but they had greater difficulty recalling the information 20 minutes later.
To wrap up our research, we talked to an addiction counselor from Recovery Happens, an outpatient treatment center. He said something extremely important, and that is that people typically drink to cover up problems and negative emotions in their life. Those who continually mask their feelings will never learn how to deal with challenges life throws, and that's what ends up getting people in the end. It takes a long time to learn how to deal with insecurities, pressure, and other social realities. You must learn to cope in your adolescent years without the influence of alcohol.